So hello everyone, good afternoon, and welcome to the FA3R webinar series. I am your host today for this session, and it is wonderful to see so many people joining us today. Today's webinar is titled AI-Powered Tools for Advancing 3R Alternatives in Research. We are excited to discuss how technology can help reduce the use of animals in research. We will explore two free innovative tools that use AI to assist researchers in finding alternatives to animal experiments. So I am happy to introduce and thanks our two experts with us today. It's Mariana Neves from the German Federal Institute for Risk Assessment. Mariana will discuss the SMAFIRA web tool, which facilitates the rapid discovery of alternative methods by sorting and ranking scientific studies. And Wynand Alkema, professor at Hans University and CSO at TenWise. Wynand will present FreeRanker, which helps to find non-animal methods for research without relying on specific keywords. We hope this webinar will enhance your understanding of how you can simplify the search for alternatives to animal use.
Thank you, Mark. Thank you for the invitation. And good afternoon, everyone. So, as Mark already said, I'm going to present our SMAFIRA tool for searching the scientific literature for alternatives. So this was a work together, my colleague Daniel and me. So Daniel is a biologist, and I am a computer scientist. So we shared the work. I did the prototype and the machine learning. Daniel did the annotation and supervised many of the functions that we implemented in this tool. So this is the agenda of my talk. So I will start with a very short introduction about the tool. Then I'll show the demo online live. And then I will talk very shortly about the methods and in our current work, what we're doing now to improve the tool. So what's SMAFIRA? So it's a free online tool to screen the literature database, right now just PubMed, for candidates health alternative methods. So it employs artificial intelligence and human feedback. As I will show, we have some predictions using machine learning, but we also allow users to use their feedback to improve the results. There is no login, so no cookies, no sensitive information. We don't get anything from the user side, except your input, of course. You don't need to enter terms for your search, so we use something else, as I will show shortly. And we make no judgment on the suitability of the retrieve methods as alternatives. So we just give some candidates, we just give some suggestions of methods, but the final decision on whether this method is an alternative or not is, of course, reserved to the human experts. So this is the basic workflow of SMAFIRA. So here we have the database with the paper. So it's PubMed that we query online, live on the flow. So the input to SMAFIRA has always a reference article. I will talk about that in the next slides. And so it's a PMID in PubMed. So from PubMed, we get this related or similar article. So if you use PubMed before, so maybe you are familiar to this similar article. This is like a precompiled similar articles in PubMed. So most of or many of the entries in PubMed have these similar articles. And then we get those similar articles. So they are already as the name says, very similar article to the one that you entered. And then we do some classification automatically with our machine learning. We detect the methods contained in the proposed in this abstract. And then we show them as a list of results in our tool, in our web interface. So we decide to use this reference article. So the idea is that you already have like an animal experiment that you are currently doing, or that you found that when searching the literature, and you want to check whether there is already an alternative method for this particular animal study for this same research goal. So this is just one example of you have a reference article. So yes, you need to go first to PubMed, and you will search for the article that you maybe already have, or search for one. It doesn't need to be an animal experiment, but we assume that usually it is one. And then you just get the PMID. So this will be your input to SMAFIRA. So now I will go to the tool. So the URL is here, and I can maybe post it in the chat later after my talk. So this is where you will going to come when you enter. You can also put SMAFIRA in Google, and Google should find it. So you can start here by the start button, or go to the search menu here. Okay, so here we're going to add to input the PMID. So I just tried the one that I was showing you, but you can just write or copy paste here, and then you click here in the search PubMed for similar articles. And yes, it didn't work. Of course, why should it work, right? At the first time during a demo. So first we get that PMID from PubMed, and the tool just confirm that you have the right article. You can also click here to go to PubMed and do your search. So when you're okay with this, and with this input, you just click on this green button, and then you go. So after clicking in this button, SMAFIRA gets those similar articles, do the prediction, and then show the results. So this is the results page. So the typical list of results. You can get different number of results per page, or navigate the pages. As I said, we get those similar articles from PubMed. So for this particular one, we have 86. So the number varies a lot. It can be even 1,000 or something like that. So as I said, there is no login. So we keep results, I would say, right now for 3 months or so. So if you want to come back to your results, you should bookmark, save somehow this head URL of your section, but this is the one that we see here, or here in the browser. And at the end, you can also download, export your results to a tab separated value format. So okay, so we have here our results. So you will see each result here at the title of the publication, and the labels here. These are the labels that were predicted with our machine learning. So here at the top you have features. So the ones that you have this red icon here, it means that for this particular set of similar articles, there are no articles with primary cells and tissues. At least that's what our machine learning predicted, or immortal cell lines. But we have some invertebrates. So if we click here, we will only see the articles. So 29 articles with invertebrates. And yeah, so you can play around with the feature. So just one with Vitrum models. And we have three types of ranking. So you have the original one from PubMed. If I click here, it's the original one as PubMed, similar article has available in PubMed. And if we click here, we have the one from SMAFIRA, sorry. So yeah, there is some mistakes. We have some maintenance right now in the institute. So that might be the reason. So if we click in one of the articles here, we go to this side-by-side comparison page. So on the left side, you have your reference article, and on the right side, you have your similar article, one of these candidate a similar article. So the idea here is that you check. You can also change the predicted label. So maybe you say, oh, the machine learning did it wrong. It's not others as predicted here. It's actually something else, and you can just click, yeah, somehow. Sorry, as I said, we have some maintenance yesterday and today, and some of the, okay, it worked here. I could predict, I could click home primary cells. And you can also compare the article. So the idea is that you can rank the article based on your feedback. So I read about abstracts, and I think that they are like somehow similar. So I click here on this label here, and you will see later you can rank the article based on your feedback. And then you go to the next article. So you read it again, and you say, okay, these two, they are not similar. So I will click on the red one, and maybe the third one, I go on, and yeah, and sometimes it doesn't work. So let's go back to the list of results, which is, oh God. So I wish I had saved I have some other results here. Let's check if I can make this one work. Yeah, so suddenly, but I have some screenshots here, just in those cases when the tool, like right now, is not working because of the maintenance. So as I said, you can compare the results, give your feedback, and when you go back to the list of results, this is what you would see, the feedback that you entered during your validation. And if you click on this My Rank, you will be able to rank the article based on your feedback, not only on those other rankings, the one that we developed and the one from PubMed. So this is basically how the tool works. So you can export results also with your feedback, and you can save the URL for later for doing it, for continuing your validation the day before, or even sharing with colleagues and do them together. So I will just shortly talk about the methods behind the tool. So a couple of years ago, we published the method behind this prediction of the label. So we annotated a corpus. So what's a corpus? So it's a collection of abstracts that we retrieved from PubMed. So we manually checked, and we have like 13 people, our researchers in our institute doing this annotation, at least 2 or 3 of them per abstract, just had to compare if they had the same opinion. So as I said, we had 1,600 abstract, and these were the labels that we annotated. So the one that you saw in the tool previously. So this here on the right, you have the agreement using this kappa agreement. So you can see that the agreement was sometimes perfect for archinvertebrate, but also very high for the other labels. But it's not perfect because people have a different opinion sometimes regarding the methods. Most of the articles have only one label, as we see here, 800 something, but Many articles have more than one label, because people sometimes propose more than one method in an abstract. So what's behind our model? So we used the transformer. It's like neural networks pre-trained on PubMed BERT. So I think that nowadays you see a lot of this talk everywhere about this large language model. So PubMed BERT is a language model. It's not that large in comparison to what's behind a ChatGPT or the new models that a large language model that you see nowadays, but it's a language model that was trained, as the name says, in PubMed. And so you can say that this language model has like some pre-knowledge on PubMed, on this biomedical domain already, and we just fine-tuned. So we just trained it again with our data, just that this model can really learn how to do this task, how to label each abstract with our 8 labels that we defined. So this is like a 2-step. So the first step is not done by us. We just get this model, which is available from Microsoft Research, and we just fine tune it for our corpus. And so these are the results. We tried different language models. So we chose PubMed BERT, the one that got better results. And this is what you see here. So as closer to 1:1, the better. So at the highest values were obtained for the invertebrates label. So this is now just a couple of comments about our current work. So we already are working on a new corpus, so like kind of an extension of this goat hamster corpus. So this time we got a 3,000 abstract. We try to get like a variety of different disease categories using mesh terms. So like to cover different disease, because sometimes depending on the disease, the type of models of animals that I use are different. We also have some additional labels. In particular, we try to split some of the labels. So a human right now is available in the tool include all human materials. And this time we try to split it according to the primary mortal cells, organ tissues in clinical studies. And we are already getting some promising results with a mix of the previous corpus and this new corpus. And at some point we'll put it on SMAFIRA, so change, of course, the interface and add these labels. We are also right now organizing what we call the SMAFIRA share text. So for those, for the ranking, we want to improve our ranking, and for that we need some data to evaluate which ranking strategy works better. So we are asking for people to annotate some reference article for us. So just the top 20. So what I just did now. So I read this pair health abstract and evaluate with those 3 thumbs up, thumbs down, whether the research goals are similar or not. And this is what we aim to use for training our model, or at least evaluating how we're ranking better. So we ask participants for doing a manual annotation using the SMAFIRA tool, or to develop system and do this annotation automatically. So we select some PMIT, some reference articles. So as I said, you go to SMAFIRA, you annotate the top 20, and then you send to us either the exported file or the URL that have your sessions. So if you're interested in helping us, just check our share task. So we plan to do a couple of publications with the participants that join us in the annotation. So in a conference for computational linguistics in Vienna in August, but also a joint journal paper at the end of the year. So please contact me if you have interest in like making a team and join us in this annotation. And that's it from my side. So thank you for your attention, and I'm happy to get your questions.
So I want to present. So thank you, Mark, and others for inviting me to give a little overview of 3R Ranker, the tool that we developed. Thanks, Mariana, for already introducing some concepts of AI and PubMed and article ranking. I will build on those a little bit more during my talk. What I will do is I will first describe a little bit the technology behind our tool and then show how we want to make this available. So I take the reverse order of the previous talk. I will start with our journey towards this tool. And it was really guided by a question that we heard a lot from our colleagues and our collaborators. How can you identify 3R related literature in books, in papers, well, in fact, in any kind of text, so also gray literature. So how do we find information on 3R? Isn't there enough information? Is there too much? Or don't we just have the correct filters to filter out the things that we want from this enormous amount of literature that is out there? And just to show the problem, and we did this together with a PhD student, Judith van Leijk, who really investigated how people go about this. There's a lot of information if you want to go and search for 3R related papers in a lot of databases that are either very comprehensive or really tailored to a certain research field, or Google Scholar, which is really area agnostic, just as indexing. But going through all these databases, having the correct search terms, using the correct key phrases is very elaborate. And we also see sometimes that links get stale and outdated. So these databases are hard to maintain and to update. And there is plenty of databases out there. So this is an interesting overview of the JRC. There's so many data catalogs that are out there that you can search. But in practice, that is what Judith also found in her PhD thesis. She said researchers find it hard to do a good 3R search because what are the keywords, where are the sources, and also, maybe most importantly, often people provide very interesting methods or test methods, and they describe that in papers, but they don't label them as a replacement for an animal method. They just describe a very good in vitro assay, for example. So you will not find those articles when you search with 3R terms or with terms like organoids, etc. So it's very hard if you go into PubMed or another database to really get an unbiased, updated, complete search if you have limited time and budget. So and that leads, in fact, that a lot of the interesting information that is out there is not used or underused. So we thought, how can we approach this problem? And in our approach, and that echoes a little bit what SMAFIRA is also doing, is we said, let's connect to a database that a lot of researchers use, PubMed, and try to as much as possible follow that workflow and that data source, and also try to make this resource automatically updatable, because we don't want to make a one-off solution that is not up to date with current developments. So we started to use PubMed for this. We published 2 articles on that already. You can have a look at those if you want to have more information on this. So the left one over here at the bottom is a more abstract description on the approach, and the other paper over here is the actual implementation of the 3R Ranker algorithm. So I will focus on the 3R Ranker algorithm and very shortly describe, very high level, how we created the 3R Ranker algorithm. And basically we followed a very a common machine learning approach. So this is a little bit different than the related papers approach that was described earlier. What we have said is we take a set of known 3R related papers. So you can do that in various ways. One of the ways we said, let's go for the mesh term that says alternative animal tests. So I think it was already mentioned that some people have different definitions on what is really an alternative test and what is really NAM or 3R, but for now we use this definition. And we take a random set of papers, the red one here, and we let the computer figure out what is the main difference between those papers. What are the words, the phrases? We also use these transformer models that Mariana described. And basically you train the model to produce a prediction algorithm, which is just an algorithm, a piece of computer code. And once you have that, you can run that piece of computer code on any paper, any text, in fact. It doesn't have to be from PubMed, but on any article. And the algorithm decides this article or this abstract title combination is 3R related or it is not. That is in very high level how we train the algorithm. And as I will show later on, this was a, let's say, foundation model. So this was the starting model, and we later fine-tuned this towards algorithms for skin alternatives and brain research, so CNS. Just looking at the time here. So what we then did, I'm just going one slide back, we said, let's take this algorithm that we produced over here and run the prediction on the entire Medline database. So we download the entire Medline database, we run our algorithm, and for 40 million papers, I think we have about now 38 million papers, we have a score that says this paper is 3R related or not. And this score is a score between 0, so totally not 3R related, very sure, don't read this, and a score of 1, which is highly probable related to 3R. So a score between 0 and 1, and based on where you put your threshold, you can have more or less papers that you classify in the green box. So this I just explained. We have 40 million PubMed abstracts with a 3R score. And the nice thing is that we do this fully automated and we download incrementally PubMed each day. So we are always up to date. As new abstracts and titles come in, we directly score the abstracts with our classifiers, and they receive a score and go into the database. So this 40 million abstracts, PubMed IDs, we have in our database with a score, and you can query those abstracts. But if you have this in a database, that's not enough to get it to you guys or to other people that want to use it. So we thought about how do we make this available? And we have two ways of doing this at this moment. We have, for people who are more into computer science, we have an applied programming interface. I will show this very briefly. So you can basically let the machine access our database and do stuff with the data that we have. And we have a searchable web interface with about, and I'll explain it later in detail, and you see a picture on the right-hand side, with about 500,000 predefined keywords that we found in these papers that describe 3R. And these keywords, they describe genes, pathways, diseases, drugs, pharmaceutical products, etc. So a large vocabulary that you can use to search these papers. So there's still searching involved, but not searching with 3R terms. That is very difficult, because what is a 3R term? But only searching with the terms that, well, represent probably your biological question of interest or the process that you're studying. So I thought this is AI in the webinar, so I thought I was allowed to show a little bit of code here, but I'm not going into detail here. What you see here is a Python script of about 25 lines, so a very simple script. And what this script does, it connects to our servers at TenWise, and it says, give me some data that you have for the 3R scores that you have been calculating. And the important part is here, the PubMed identifiers, and it was already explained in the first talk. These PubMed identifiers are 7 or 8 number digits, and you just say to the script, I want to have the prediction for these papers, and you send this message to the computer, to our servers. And what you get back is the PubMed identifiers, the classifier that we use to classify the abstract, and the score that is associated with it. So it's a very simple output. And what you get is, I showed it here in Excel, for a number of PubMed identifiers, you have the model that we developed and the, I don't see it, I have to move this window away. Oops. Here. And you see the score. And as I already indicated, the higher the score, the closer to 1, the more confident we are that this article is really interesting. So this is doing this with scripting. We apply this, we supply this for free. So if you want to have these predictions in your own tool, you're more than welcome. Just send us an email and we'll set you up, and you can just generate your own data for the PubMed identifiers that you're interested in. So these are the numbers here. It was already explained, the PubMed identifiers. If we look them up in PubMed, well, we already see based on the title that well, at least to me, they are very interesting, because it says talks about in vitro assays. It says cellular models, and here we talk about the induced pluripotent stem cells, which we a lot see, by the way, in neurodegenerative disease research. So a high score corresponds, and we validated that and did that with experts to well, we hope a very interesting article that people can use to read about their essay, set it up in such a way, incorporate it in their research, or suggest it to others to use it. So this is keyword free. We just have, for all the PubMed identifiers, we have this score, and it's available. We have a look at the time. Yeah, I'm okay. So now I want to go to a demo of the web tool. That is the second thing we developed. So we first started with the API, and then we went to the web tool. Let me try to go there. So the web tool is over here, just refreshing. What you can do in the web tool is to, I will just start typing. For example, if I'm interested in Parkinson's disease as a research topic, I will type in some letters of the concept name. So this is the keyword search with, let's say, the biological interesting question that you may have. And you will see the terms that we have in our database. Don't look at the identifier, it's a bit boring. Look at the concept name, for example, Parkinson's disease. And if we click on this, we directly see the abstracts on the right-hand side that have abstracts that deal with well, alternatives in relation to Parkinson's disease. So we see stem cells, we see in situ detection, multicellular 3D units on a chip. I'm not familiar with all of those, but I think they represent interesting papers. But what you see here, I should go a little bit slower, is that we have scores for 2 classifiers, the one we developed for skin and the one we developed for CNS. And we see often that both classifiers may sometimes give a high score for the same paper, because a lot of the terms like in vitro and cellular model and 3D reconstituted organ are valid across multiple disease areas. But let me go to the specific CNS classifier. So we will see the score over here, and we can sort on the score, and the higher the score, the better it is. We can also look for publication year. And what is interesting is that the last article, at least that we score, has been published in 2022. This is not the last article on Parkinson, but the last article that we think is 3R related in relation to Parkinson's disease. And of course, you can argue you should have more papers. It depends on your definition of what is 3R or not. And we are rather restrictive in that. So we limit our database a little bit to not have too many articles in there. So we have a few 10,000 in the database at this moment. You can also search to see if you're interested, for example, in astrocytes, you directly filter the table to only have papers that have astrocytes in them. And whenever we have a PDF, we link to that, and we also link to the PubMed identifier. So you go directly to the PubMed identifier over here. So this is searching with a disease. You can also search, for example, I had this dermatitis example in my slides, and now it says, hey, dermatitis is in our database, but we don't have a hit with the CNS classifier. You may want to use the 3R skin classifier. And we take the 3R skin classifier, and we directly get high scoring papers there for dermatitis. And we also highlight a little bit wherever we have the term in there. So this is entering the database with disease, but we have many keywords that we attach to that. So I could also say, for example, I'm interested not in a disease, but my favorite gene is this gene. I'm studying this. I used to work on P38, which is MAPKAP-K14. And now I directly go to studies where this gene is mentioned in the abstract. So for example, we have here a reconstructed human epidermis. And now I have to look for P38, of course, in this abstract, but I trust it somewhere there. Yeah, it's over here, P38. So you can not only go with the disease, but also with the gene. Well, in fact, any word that we have in our database. For example, another one that is interesting, because I already checked that, I need to, of course, type it correctly, is autophagy, which is a very important pathway. It is related to clearance of proteins that are not functioning anymore. So if I click on autophagy, I get papers that still are about autophagy. It's over here, but it's safety assessment related alternative testing strategy. So now we go in with a pathway, with a more broad concept, and we get articles that are interesting, or at least that's the idea, that they are interesting. But we tested that with a number of experts, of course. Also here we can score on year, and here we have one for 2025, just to show that we are rather up to date here. So this is kind of how it works. I can do many more examples, but in view of the time, I will not do this. But it's, yeah, I hope you get the gist from what I've been showing here. The other thing, I think the same as was said in the previous talk. We are looking for extension of this database with new classifiers. So we also have a screening system in place where we have a number of reviewers. In this particular case, we have 2 actives on this particular site that are presented random abstracts for a certain disease area and do the scoring to say no 3R, maybe 3R, and 3R. And we use that input to update our models or to create new models for new disease areas. I think I will leave it for now for the demo here, and go back to my slides. I'm not seeing any. Oh, yeah, slides. Oh, now I have to. Whoop, because I also made screenshots, of course. Good practice to do this. So I will end with 3 more slides, and the one is that I would say the take-home messages. This 3R anchor approach. Well, what is important, we are extending this towards new research fields, just as was shown for SMAFIRA tool as well, new disease areas or other product areas, whatever you want. it can be incorporated in other tools via the API. So we invite everybody who wants to incorporate our predictions in their application, like for example, SMAFIRA tool. We're very happy to assist in that and to give you access. We update this automatically with new PubMed data. And the interesting part, I think, that I did not yet dwell on too much is that it can be applied towards new text sources, because we have a model that is capable of scoring any kind of text. We just use it on PubMed, but it can be used on any kind of text. So what we did the other day is to, or no, it was last year actually, to do a complete scraping of the Science Daily website, which is kind of a new site and new developments in science and what companies and universities are doing. And we just run our algorithm on those websites. It doesn't have to be an abstract, as long as we have text. And just to give you an example, a news item about oil seeds, crops waste for protection of skin from sun, so for sun blocking. We find that this particular news article has a high score on the 3R skin classifier. And if you look into the text of this website, we see the 3D skin reconstructions, and there were more terms like that in the narrative there. So we can use this algorithm also for websites, but also for, I don't know, governance documents, meeting minutes. We can use it on any kind of text. So it's in that respect, it's very versatile. So we also make our algorithm available for these kind of applications. So just send us a message and we will help you to get started with that. But you have to do your coding then yourself, of course, to get it working for new text sources. So I want to end my talk with two outlooking statements and flanking research. On the left-hand side, I'm posting a picture here from a LinkedIn post from Freshie, from the company Freshie. Together with them and a few more companies, we are with the JRC working on a similar database where we use similar technology, AI, for the detection of human biomedical models. So this is a little bit broader than our strict 3R terms definition that I used before. Same technology is more or less used, but the database is much bigger. So we're working hard on that. I think this will be available in at least this year, but I'm not going to put a real date on that at this moment. But it's a very interesting project to work on. So if you look for Freshie or TenWise, you will see more information on that. So that's one thing that is a nice spin-off and a nice combination of the research that we are doing. The other thing is an interesting consortium that we are working in, but it's more geared towards not researchers, but more to the regulatory and the regulatory side of safety testing or animal testing, and especially the reduction of animal testing, I should say. And we are working in that project on an ontology for specifically, well, maybe 3R is too narrow. NAMs could also be a good term here, but what are the assays, what are platforms, what are cell types, etc., that are typically being used in this field of research? How do these terms relate? Because if we have these vocabulary terms, we can much better tune our machine learning models with this knowledge and give more weight to the words in the text if they correspond to a word of this vocabulary. So those projects are ongoing, and they all have a little bit different aims. So I don't think they replace each other, but they really complement each other. So hopefully you will see output from these projects integrated into 3R Anchor and the other way around, of course. So with that, I want to thank you, the people who worked on this. from Karolinska, from the SAFE consortium, from TenWise, and also the Swedish body that supplied funding. And we also got funding for this from the province in the Netherlands that makes this development possible, and makes it also possible that we can release these results and this service free of charge to people who want to use this.